Okay, before I start, I want to give a reminder that mitosis is not actually a cell division. It's actually just a nuclear division. I'll explain more on that later, but it's something to keep note about. Okay, cell division is what is when a cell divides into to produce new cells, and there are two stages: karyokinesis and cytokinesis. Karyokinesis is when your nucleus divides. So there are two types. There's mitosis and there is meiosis. So yeah, so when the nucleus divides, that is karyokinesis. Cytokinesis is when your cytoplasm divides. The organism's body cells are divided into somatic cells and reproductive cells. So somatic cells usually go mitosis, and they are the cells that you normally see around your body. And they are a diploid number of chromosomes, which means double the normal set, which is 46 in humans. For gametes are reproductive cells, and they contain a haploid number of chromosomes, which is 23, half of a diploid. So they are produced through meiosis. So the reason why why not call N46 and N over 2 23? Because actually the 2N here shows that one N comes from the mother and one N comes from the father. So we have 23 cells from the mother and 23 cells from the father. And we call those maternal and paternal chromosomes. And some of the chromosomes share similarities. So a mother and a father chromosome that shares the same characteristics are called homologous chromosomes. Another thing that I wanted to keep in mind is chromatin, chromatid, and chromosome. Chromatin is a string-like band, a thread-like thing, and it is not visible under the microscope. While chromatid can also be like this. This is visible. So yeah. So but then the thing is, both of them are still chromosomes. So if it's a chromatid and it's a chromatin, you have to know when is it a chromatid, when is it a chromatin. But in the end, they're both still chromosomes. So cell cycle and mitosis. What is a cell cycle? It is a sequence of events that involves DNA multiplication and then cell division. So DNA multiplication is interphase, and cell division is mitosis plus cytokinesis. So interphase is divided into three: G1 phase, GS phase, and G2 phase. For G1 phase, they grow their main components such as mitochondria and endoplasmic reticulum. You know those stuff that you need in a nucleus. And then they also produce some proteins. And these all happen in the nucleus. So during G1 phase, the nucleus expands, it becomes bigger. And during this time, chromosome is still chromatin. S phase is where the DNA starts to replicate. So S phase is where the DNA starts to replicate. And this causes the chromosomes to multiply as well, because the DNA is inside the chromosome. So when the DNA inside the chromosome replicates, it becomes 2. And these two identical chromosomes are called sister chromatids. But since they're replicated together, they are still connected to each other through the centromere. And again, since they're both identical, they both contain the same DNA molecule, the same structure, the same genetic content. So this is the most important part. You have to know that S phase is where a DNA gets replicated. G2 phase is where your cell continues growing and it gets ready to enter the M phase. The M phase is divided into two, which is mitosis and cytokinesis. Take note that most of your cell undergoes interphase. Mitosis and cytokinesis is just a very tiny part of the whole cell cycle. So mitosis is the division of the nucleus into two nuclei. And before mitosis, it's 2N. After mitosis, it's still 2N. So how does this happen? So during prophase, these chromatin are already connected to the centromere, right? So you can now see them under the microscope because you cannot see them under the microscope because they already turn into chromatids. They are shorter and thicker, so it becomes easier to see them through the light microscope. And again, as I said, they're both joined at the centromere. And during prophase, the nucleus disintegrates. The nuclear membrane and the nucleolus disappear. So this and then the centriole appeared during the interphase. They move. There are two of them. Each of them go to their own separate poles. And then these centrioles start to have spindle fibers, and the spindle fibers connect to the centromere. So what happens is, when the spindle fibers connect to the centromere, it moves the chromosome to the equatorial plate, and this is when metaphase starts. When the centrioles are already at the opposite side of the cell, and the spindle fibers and the chromosomes start moving to the equatorial plate. The spindle fibers make sure that they stay there. So now they're all at the middle. The equatorial plate can also be called as the metaphase plate. And then interphase is basically just the chromosomes all aligning in the middle. Anaphase is when they spread up. The way they spread up is the centromere divides into two. Why does the centromere divide into two? That is because the centriole triggers the spindle fibers to contract. When the spindle fibers contract, left contract and right contract. So the centriole cannot handle it anymore, so it just splits into half. So one half is carrying one chromatid, so now the sister chromatids are separated. And then anaphase ends after the chromatids reach the opposite poles of the cell. During telophase, the chromatids are already at the opposite ends of the cell, so now they're called daughter chromosomes. And each pole contains a full set of chromosomes. So if you have 46 here, you also have 46 here. And I'll get to why later. Chromosomes are shaped again, okay, and then chromosomes turn back into chromatin. After that, the nucleus starts to come back. It forms again, nucleolus and nuclear membrane. And then the spindle fibers also disappear. And after that is cytokinesis. So cytokinesis occurs in both plants and animals, but the main difference is plant cells do not actually have centrioles. They do not have this thing. And they do not have the centriole. The ones that at the polar. But then the plant cells do have spindle fibers, so they can still split up. So cytokinesis is different between animal cells and plant cells. Cytokinesis happens immediately. Cytokinesis happens immediately after telophase. In animal cells, the plasma membrane constricts in the middle of the cell between the two nuclei. What this means is, this is the plasma membrane. This is the plasma membrane. At the middle, they start to contract. They start to split up. They start to cut in the middle. So why does it cut in the middle? Because this thing contains microfilaments. And the thing here is called the cleavage furrow. So at the cleavage furrow, the microfilaments start to contract. So when the microfilaments contract, it causes the cleavage furrow to contract as well, like the ring. The middle starts to contract, so it becomes like an H shape. I mean H shape, yeah, H shape, definitely H shape. So so at first it's like this. And then the microfilaments here, they start to contract. And when they contract, they pull it like this. So it becomes an H shape. And then, And then it will keep doing that until it splits out. For cytokinesis in plant cells, the vesicles form cell plates at the center of the cell. So the cell wall contains vesicles. And then, the plant cell contains vesicles. And then they form cell plates at the middle. And then this cell plate is surrounded by a new plasma membrane. And then at this plasma membrane, it will form a new cell wall. So this cell plate, it starts from here. And then it gets bigger and bigger and bigger until it completely covers it. And then after that, this is here. It expands outwards until they all combine with the plasma membrane. The plasma membrane is basically the outside of here. So it will expand until it attaches to the whole cell. And then at the end of cytokinesis, cellulose fibers produced by the cells to strengthen the new cell walls. Two daughter cells are formed. Each cell has a diploid condition. Okay, so after cytokinesis, instead of splitting like in animals, they split. They actually just stick together. Instead, they form like continue forming like this. And so they just keep forming and then they keep forming like that. For animal cells, it splits, right? But in plant cells, it actually stays. So this is one daughter cell, this is two daughter cells. This is a cell wall. Because remember, cell wall are all connected in plants. So you don't have to split up the plant cells. You just have to make sure that there's a cell wall in the middle of the two nuclei, and then there are already two different plant cells. Okay, now the main question. I have 46 chromosomes here. I have 46 chromosomes here in the normal cell. And then they divide into 2. Wouldn't it become 23 if I divide into 2? Now, that's where you have to consider. During S phase, the chromatid duplicate to become sister chromatids during S phase. So when they become sister chromatids, one chromatin becomes 2 chromatids. So one chromatin becomes 2 chromatids, but they are both one chromosome. This is one chromosome. This is one chromosome. It will not be considered as 2 chromosomes until what's it called again? The centromere gets split up, which is only after anaphase. So only during anaphase. So these sister chromatids, even though there are 2 chromatids, and it is still considered as one chromosome. So after S phase, there are actually 92 chromatids, but there are only 46 chromosomes. And then these 46 chromosomes go to the middle, and then since there are 92 chromatids, 46 chromatids go to the left, 46 chromatids go to the right, and now these are 46 chromosomes, and these are 46 chromosomes as well. So that's the main thing you have to know about mitosis. Why do we need mitosis? First of all, embryo growth, because you can't grow without mitosis. Regeneration of certain parts of the body, such as the lizard's tail. Produce new individuals through asexual reproduction. Replace cells that are damaged or dead. There's also other stuff, but you don't have to mention much. Okay, meiosis is a process of cell division that occurs in reproductive organs to produce gametes that contain the half the number of chromosomes. So the main difference between mitosis and meiosis is in mitosis, one cell produces 2 diploid cells. In meiosis, one diploid cell produces 4 haploid cells. So why do you need meiosis? Because when you want to reproduce, you need the sperm and ovum to fuse together, right? So the sperm and ovum, if the sperm and ovum fuse together and they both have 46 chromosomes, when they combine together, wouldn't that make them 92 chromosomes? So you don't want that to happen. So instead, you want it to be half. So 23 from the father, 23 from the mother. So in total, you have 46. So that is why you need meiosis. Other than that, during meiosis, there will be some processes that will cause the genes of the chromosome to change slightly. So that is genetic variation. Meiosis is divided into 2. Meiosis 1 and meiosis 2. The main difference is, in meiosis 1, between meiosis 1 and meiosis 2, cell cycle only happens during meiosis 1. During meiosis 2, there is no cell cycle. No S phase, no G1 phase, no G2 phase. It is directly to the point. So during meiosis 1, the cells already replicated. So now you have, again, 92 chromatids, but 46 chromosomes. And similar, chromatin shortens, thickens, and then they form visible chromosomes. And then when homologous chromosomes pair together, when this is a homologous chromosome, and then there are two homologous chromosomes. If two homologous chromosomes pair together, that is called a bivalent. And a bivalent is also known as a tetrad. I prefer calling it a tetrad lah. So this pairing of two homologous chromosomes is called synapsis. So yeah, this is a homologous chromosome. Wait, no. This is a sister chromatid, because remember, during replication, the chromatin replicates into chromatids. So this is a sister chromatid. And then two sister chromatids combine together, they come together through synapsis to form bivalent. But the main thing is, these two sister chromatids, the two pairs, they need to be similar or homologous. So keep in mind, this is one chromosome, this is another chromosome, and when these two pair together, that's called synapsis, and this whole thing is a bivalent or a tetrad. This alone is sister chromatid. Two sister chromatids together that are similar, two pairs of sister chromatids that are similar are homologous chromosomes. When homologous chromosomes pair together, they are called tetrads or bivalent. And then when they are at this bivalent phase, you can see that the chromosome can cross over at one place called the chiasma. So the crossing over area is called the chiasma. And when they cross over, the genes exchange between the two non-sister chromatids. So this will create a new combination of genes. And then after the chiasma is formed, then the nucleolus and the nuclear membrane start to disappear. And then after that, then the centrioles will actually move to the sides, to the opposite. And then the spindle fibers form. So the main difference between prophase 1 and prophase in mitosis is there is a crossing over of tetrads which were formed through synapsis. In metaphase 1, the homologous chromosomes are arranged at the equatorial plate. One chromosome from each pair of the homologous chromosome is tied to the spindle fibers from one pole cell. And its homologous is tied to the spindle fibers from the opposite pole cell. So what this means is, if this is the center, the equatorial plate or the metaphase plate, and these are the homologous chromosomes, the spindle fibers are not like this. It's like this. Okay, wait. Yeah, it's like this and like this, not connected. It's like that. And the main difference between this and mitosis is, in mitosis, the centromere goes exactly in the middle. But in prophase 2, they don't. They actually, the two homologous chromosomes are still together and they're still at the equatorial plate together. And again, in here, the sister chromatids the centromere is connected by two spindle fibers. But for here, they wouldn't contract yet. They wouldn't separate yet, because the centromere is only connected to one side. So the centromere moves together with the sister chromatids. So after anaphase, they are still sister chromatids, but the homologous chromosomes will split up. So when the spindle fibers contract, it will force the homologous chromosomes with the chiasma and so on to separate from its pair, and then they'll go to opposite poles. And remember, they are still made of sister chromatids because the centromere is still there. During telophase, they arrive at the opposite cells. The nuclear membrane and the nucleolus reappears again. Spindle fibers disappear, and now remember what I said. Centromeres together count as one chromosome. So now there are 46. There are 46 of these, right? 46 centromeres. So 23 here and 23 here. So since they haven't split up yet, so there are now 23 chromosomes on the left and 23 chromosomes on the right. So they are now haploid. Okay, after telophase 1 is cytokinesis, and then it will produce two daughter cells, and these two daughter cells are in haploid condition. And during this time, there is some interphase, but there is no S phase. The DNA does not replicate. There is no replication of DNA. So the sister chromatids stay like this. They stay together. So now during prophase 2, the nucleolus and the nuclear membrane disappear again. And then now the chromosomes are still in sister chromatid form. And now they will meet the centromere, like normal. And then the spindle fibers start to form like normal as well. And then during metaphase, the chromosomes are arranged at the middle is of the equatorial plate. And the centromere is connected to both both poles of the spindle fiber. So the chromatid is connected to both poles. And then during anaphase, the sister chromatid separates because the spindle fibers contract. When the spindle fibers contract, the centromere splits, and the sister chromatids move from, to the opposite poles. So now they're called chromosomes, daughter chromosomes. And then during telophase, the spindle fibers disappear, the nucleolus and the nuclear membrane appears again. And then as half the number of parent chromosomes. So compared to the first cell, which is 46, we only have 23. But the main difference is, we already know that this is 23, right? So since there are 23 chromosomes, and they all arranged at the plate, they separate. So 23 go to one side, 23 go to the other side. So these four cells are also haploid cells. Haploid means half. And the main difference between mitosis and meiosis is the genetic content. In mitosis, the genetic content is actually the same. But in meiosis, the genetic content has already been slightly adjusted. And that is because of the chiasma that I mentioned earlier. When they cross over, there is an exchange of genes. So the genes change a bit. And that is where the genetic content changes. Okay, issues of cell division on human health. So uncontrolled cell division is called tumor. There is the benign tumor and the malignant tumor. Benign tumor is not that dangerous. It's like an extra bulge from your body. Like for example, some people might have an extra thing coming out of their face. It is not dangerous. It does not do anything to you, and you can remove it through surgery without any problem. But a malignant tumor or cancer is very dangerous, and it's because I'm sure you will know the causes of cancer, so I'm not gonna mention it. What happens is, when these cells divide uncontrollably, they actually divide in very important organs, and they develop in very important organs. So when they develop in very important organs, it ruins the environment of the normal environment of the organ. Like organs are supposed to like divide normally, but because of this cancer cell, it divides very uncontrollably, and this is very dangerous to the organ. And since the cancer cells divide quickly because of mutation, it also causes what happens is it divides way faster than the other cells. So when there are more of these cancer cells, they take more nutrients away. So when more nutrients are taken away, then the organ starts to have trouble. But then since the cancer cells took the nutrients through the blood tissues and the blood vessels, when the cancer cell accidentally goes into the blood vessels or the lymph vessels, they spread to other parts of the body. So if you have, for example, cancer in the, if for example, if you have cancer on the breast, it could spread to the kidney through the vessels. So you have to act quick before that happens. Now there is explanation to why they divide uncontrollably. Let me just look at the UEC book, because that's where it came from. Chapter 13, I think. Yeah, give me a bit. Okay, so there are two things. Proto-oncogenes and tumor suppressor genes. So these two have what are these two affect each other so that the cell division goes smoothly. Proto-oncogenes are mainly responsible for the regulation of cell proliferation, differentiation, and growth. So proto-oncogenes, they promote cell growth. Tumor suppressor genes, they inhibit cell growth. So proto-oncogenes promote cell growth. Tumor suppressor genes inhibit cell growth. So because of this, let's say in the organ, if the cell divides too quickly, the tumor suppressor genes will tell the cell don't divide too quickly. If the cell doesn't divide fast enough, the proto-oncogenes will tell them to divide faster. So these two genes control each other. They restrict each other to make sure that the division goes equally, in relative equilibrium. It goes constantly. But when a cell is infected by a virus, when the cell is infected and it undergoes mutation due to all of these factors, these proto-oncogenes, the proto-oncogenes, they mutate. So now it's no longer proto-oncogenes. It's just oncogenes now. So when they form oncogenes, when they form oncogenes, it's going to cause the division to become even faster. Because remember, proto-oncogenes help with cell growth. Oncogenes magnify that effect. So it makes cell growth even faster and faster and faster. So it's a higher than normal growth rate. And it over-proliferates. It divides too fast. So that is one type of mutation. The other type of mutation is the tumor suppressor genes, they get killed. Either because the proto-oncogenes killed them, or there's a mutation inside the tumor suppressor genes to cause the tumor suppressor genes to become inactive. And when they become inactive, they deactivate. When they deactivate, there is no gene that can control the cell growth. Since there is no gene that can control the cell growth, the oncogene just keeps on dividing and dividing and dividing, so it becomes uncontrollable. And because of this, there's so many cells that compete for nutrients. If a cancer cell succeeds in getting into the blood vessels, it will go throughout the whole body. So yeah, that's one type of sickness regarding cell division. Okay, I'm looking at the UEC book again. So the main effects of cancer is, number one, immortality. Effects of cancer cells. Immortality. That means the cancer cell cannot die. Let me see. Normally in a cell, if a cell mutates or it goes wrong, it will be told to die. The cell will be told to kill itself. So so that means that no bad cells will be alive. But in cancer cells, that doesn't happen. So because it doesn't happen, the cells don't die. So they are immortality. And then number two is migration. Migration is the one that I mentioned just now, where if your cancer cell gets into the blood vessels or a lymphatic vessel, it will go throughout the whole body. So it's migration. And last one, loss of contact inhibition. Loss of contact inhibition means normally when cells come into contact with each other, they stop growing. So when you see a normal cancer cell, a normal cell, it will be very arranged because they stop growing after they meet each other. But in cancer cells, even though they meet each other, they do not stop growing. Instead, they grow outwards. So it will go like outwards like that. So this is a cancer cell. It doesn't follow the rules. Other types of cell division problems include meiosis punya problem, which is when meiosis goes wrongly, it can cause Down syndrome. Down syndrome happens when during anaphase 1 or anaphase 2, one of the spindle fibers fail to contract. When one of the spindle fibers fail to contract, instead of it splitting for like this, the whole thing moves together. So one part has 23, the other part has 21. So one part is 24, the other one has 22. So yeah, a normal gamete has 23. But if one of these sperm or ovum contain 24 or 22 chromosomes, which is not normal, and it gets fertilized, it will carry 47 chromosomes. A normal human has 46. A person with Down syndrome has 47. And normally, that one cell that has a problem, this one spindle fiber that first function, is the 21st chromosome. So the other name is called trisomy 21. Trisomy 21. So that's the other name of Down syndrome. Just the 21st chromosome, the 21st chromosome, the 21st set of chromosome, has more than 2 chromosomes. In other words, more than 3 of them are identical, and more than 2 are identical. So in normal meiosis, it's 23 and 23, right? But if anaphase goes wrong and the spindle fibers fail to contract and the chromatids fail to separate, one will have 24, the other one will have 22, and then when they add together, it's 47. And that extra chromosome is usually in the 21st type of chromosome, because there are 23 types of chromosome. Remember, chromosomes have similar types, and the similar ones form homologous chromosomes. The 21st type of chromosome